And what it has is a heme cofactor. Cofactors are molecules that help proteins grab whatever their substrate is. And this ring-shaped molecule you see here is called a heme cofactor. Darn it. There we go. This is called a heme cofactor, and all cytochrome proteins, by definition, have a heme cofactor as part of their structure. We have some cytochrome proteins in our bodies that do various things. Hemoglobin's a good example, the one that carries oxygen, but this is not carrying oxygen, it carries high energy electrons. Okay, so this cytochrome complex right there has received the high energy electrons, and what it's going to do is use their energy to do active transport, pumping a hydrogen ion from the stroma into the interior of the thylakoid complex, or the thylakoid. In other words, it takes a hydrogen ion there and pumps it in like that. And it is active transport because it's going to keep pumping hydrogen ions in, and so you're going to get higher and higher hydrogen ion concentration inside the thylakoid membrane. And remember, that's not the direction the molecules want to go. They don't want to go from low concentration area to high concentration area. They want to spontaneously diffuse from high concentration to low concentration. So if you do something that's non-spontaneous, it's going to require energy. That's why the two high energy electrons are needed by the cytochrome complex protein. Let's watch it happen again. There goes plastoquinone. Shuttles them on over to the cytochrome complex, and again the energy is used to pump another hydrogen ion into the interior of the thylakoid. And so we're building up a concentration gradient. Let's watch it one more time. Another hydrogen ion gets pumped. And so pretty soon, you've got that. You've got a hydrogen ion gradient inside there. And you know, that's a form of stored up energy. That's potential energy. It's like water stored up behind the dam. And so let's pause for a moment to consider all the energy changes that we've been talking about so far. The energy was originally in the form of photons in the sunlight here. And then that was transformed into the kinetic energy of some electrons, and then those electrons were used to power active transport, and so now the energy is in the form of a hydrogen ion concentration gradient across the thylakoid membrane. So the energy keeps changing form, so we're not done with it yet. Our ultimate goal is to change it, amongst other things, into ATP energy. Well, that's the next step. Right here is a protein called ATP synthase, and it's a passive transport protein. And remember, passive transport proteins are transport proteins that allow solute molecules to diffuse, to go from their high concentration side to the low concentration side. So hydrogen ions can zip through here back into the stroma. But what this ATP synthase does is, as those hydrogen ions come barreling on through, it uses part of their kinetic energy to make ATP. And I think we'll see that here when I click this. Oh, it's not. What's it going on? Go back. Watch this. Ooh, there we go. Every time a hydrogen ion comes zipping on through, it generates an ATP. Let's watch it one more time. In, and another ATP. Pretty cool. In my cartoon here, I just showed the ATP synthase protein as just magically creating an ATP out of nothing, and that's not true. What actually happens is, here's the ATP synthase, it actually grabs an adenosine diphosphate and an inorganic phosphate and using the energy of the hydrogen ion zipping on through, connects those two together to make an ATP. If you could shrink yourself down and grab an adenosine diphosphate and an inorganic phosphate, it takes energy to link them. It's a non-spontaneous reaction, and so that's coming from the kinetic energy of the hydrogen ion diffusing on through. Sound all right? All right. Well, let's see. So we're partially at our destination. We've turned the light energy into ATP energy. But we haven't really addressed where the NADPH comes from, or where oxygen comes from, if that's part of this reaction also. And so let's begin to address those. When I've been showing the electrons, watch what happens to the electrons. They come out of photosystem 2, right there, and then they end up right here. But photosystem 2 can't have an infinite supply of electrons. You know, it has lots of them because it's full of atoms and covalent bonds which have electrons, but it can't keep giving those electrons away forever. It would eventually run out. And so somewhere there's got to be something that feeds electrons into photosystem 2. Photosystem 2's job is really to energize electrons, but it's got to have some electrons flowing into it somewhere. And the shorter answer is water molecules provide the electrons to photosystem 2. Let's look at a water molecule. You're familiar with the basic structure. And remember that these lines connecting the oxygen and the hydrogen are covalent bonds, each of which is a shared pair of electrons there and there. And so we could just as correctly show a water molecule like that as an oxygen atom, two hydrogen atoms, and between each oxygen and hydrogen is this pair of electrons. Now, what we're going to do next is kind of deconstruct the water molecule, take it apart into its bits. And this is sometimes called splitting a water molecule. And to begin with, remember that oxygen pulls harder on electrons than hydrogen does. And so it's pretty easy for oxygen to suck its electrons back. Oxygen owns this electron of this covalent bond, and oxygen owns that electron of that covalent bond. And so fairly easily, oxygen can be coerced to suck in its two electrons and take them back. Hydrogen, the hydrogen atoms own this electron. This hydrogen goes with that electron, and this hydrogen goes with that electron. But you might remember hydrogens don't pull very hard on their electrons. And so it's fairly easy to strip those hydrogens of their electrons. Now, if we take those electrons away, the hydrogen atom becomes a hydrogen what? Ion, right. And what will be the charge on the hydrogen ion? +1, right? Remember, you have an atom, you take away one electron, it gains a +1 charge. And so that's what we're going to see here. Those electrons can be removed from their hydrogens, and you get a hydrogen atom. Hydrogen ion. So one water molecule can be split apart into one oxygen atom, two hydrogen ions, and two electrons. And that is called water splitting. And ultimately, it is the source of the electrons that flow, that go into photosystem 2, that feed electrons into photosystem 2. Oftentimes, if you read textbooks, this is how they show the water splitting reaction for feeding electrons into photosystem 2. But it's slightly inaccurate, and here's why. Single oxygen atoms, like they're showing there, don't really exist. Oxygen likes to pair up with other atoms if it can. And so a better way to show it is this. If you imagine a second water molecule being split, okay, so there's the equation for a single water molecule being split. If you imagine a second water molecule being split, you'd end up with two oxygen atoms, 4 hydrogens, and 4 electrons. And what would happen then is these two oxygen atoms would then bond to each other, like this, forming a molecule of O2. And that's what actually happens in the water splitting reaction. So this is the more correct way to show it. For every 2 water molecules that get split, you get a molecule of molecular oxygen, O2, and 4 hydrogen ions and 4 electrons. And this is the source of the oxygen in the photosynthesis equation. Oh, actually, I'll show you the equation again in a second. So there's photosystem 2. And underneath photosystem 2, sticking down into the interior of the thylakoid, sticking down into here, is an enzyme that's capable of splitting the water molecules, this purple-looking thing right here. So when photosystem 2 gives up a pair of electrons, like we're going to see here, so there they go, get stuck in the reaction center, and then plastoquinone comes along and takes them away. Oh, here's a review question. Plastoquinone is going to deliver them to where? To what? To cytochrome, right, complex, which is going to pump hydrogen ions. Anyway, so plastoquinone takes them away. Now those electrons have to be replaced. Well, so a water molecule comes along. It's going to bind to this water splitting enzyme right there. It's going to get split. The 2 electrons go up to replace the ones that were just carried away. The hydrogen ions are just going to diffuse here to the inside of the thylakoid, and the oxygen atom is just going to temporarily hang out for a moment. Now, another 2 electrons, maybe the same 2 electrons, get re-energized, get taken away by plastoquinone into the electron transport system. And to replace them, another water molecule gets split. And there's that second molecule of oxygen. It will join with the first molecule of oxygen to give you O2. And that's the water splitting action. So that's where the oxygens come from in photosynthesis. There. Here we go. If you look at the photosynthesis equation, remember it looked like this. Plants take 6 molecules of carbon dioxide from the air, 6 molecules of water from the ground, and carry out a chemical reaction to change the